Medicos, welcome to the channel page. So I'm excited to be here and I hope you guys enjoy what we have to share. Today's topic is erythropoiesis. Before going into the topic, let us discuss about RBC's function. So RBCs acts as carrier to transport oxygen as well as carbon dioxide, and it provides viscosity to the blood. So, today's objectives: introduction to ABC, site and stages of erythropoiesis, regulation, factors, and some of info about reticulocytes. RBCs is also called as erythrocytes. RBCs is one of the important cellular component of the blood. Each RBC is bounded by a cellular membrane, and its cytoplasm contains hemoglobin pigment, which is a quaternary structure of a protein which gives red color appearance to an RBC. And it lacks nucleus and it lacks cellular organelles such as mitochondria, and RBC's source of energy is dependent only on glycolysis. And biconcave shape of an RBC is maintained by two proteins called as spectrin and ankyrin. As far as biconcave shape is maintained, it resists hemolysis. Diameter of an RBC is 7.5 micrometer and its thickness is 2 micrometer, and mean corpuscular volume, that's MCV, represents RBC cell size. It ranges from 78 to 94 micrometer cube. When it increases beyond 94 micrometer cube, it is called as macrocytosis. When it decreases below 78 micrometer cube, it is called as microcytosis. And normal count of an RBC in adult male is 4.5 to 5.5 million cells per mm cube. In adult female is 4 to 5 million cells per mm cube. In infants, it is increased such as 6 to 8 million cells per mm cubes due to because of hemoglobin F, that is fetal hemoglobin. When RBC count is increased beyond the 7 million microliters, it is called as polycythemia, and in pathological condition it is called as polycythemia vera. And physiological conditions, we can see increase in the RBC count in high altitudes and in hypoxia condition in excessive exercise, and you can see decrease in RBC count in pathological conditions called as anemia, and in physiological conditions such as pregnancy and hypertension. Normal lifespan of an RBC is 120 days, after which it is destroyed or hemolyzed in spleen, which is called as extravascular hemolysis. The sites where mature RBCs are produced are in intrauterine life for the first 2 months in yolk sac, which is also called as mesoblastic stage, and in third month in liver and spleen, and at 7th and 8th month in bone marrow of vertebrae and long bones. From bone marrow, hematopoietic stem cells are produced. Hematopoietic stem cells have two fates, which can differentiate into myeloid progenitor cells and which can differentiate into lymphoid progenitor cells. Lymphoid progenitor cells further differentiates into T lymphocyte and B lymphocyte. Myeloid progenitor cells further differentiate into erythrocyte and other leukocyte and platelets as well. Coming to stages of erythropoiesis, the first stage is hematopoietic stem cell. Its size ranges from 19 to 23 micrometer, which can self-replicate and differentiate into progenitor cell. And next stage is common myeloid progenitor, which acts as progenitor for erythrocyte, leukocyte, and platelets. For except lymphocyte, it acts as lymphoid progenitor cell. And the next stage is proerythroblast. Its size ranges from 15 to 20 micrometer, with 2 to 3 nucleoli are seen, and hemoglobin is not formed at in this stage. And next stage is early erythroblast. Its size ranges from 12 to 18 micrometer, and hemoglobin is synthesized in this stage. Coming to next stage is intermediate erythroblast. Its size ranges from 10 to 15 micrometer. Nucleoli is disappeared, and heterochromatin is clumps inside the nucleus, which is pycnotic nuclei, and hemoglobin is visible under light microscope in this stage. The first three stages are basophilically stained because of nucleus, and from intermediate erythroblast onwards it is stained by eosinophilically stained because of progressive loss of nucleus. Intermediate erythroblast is stained by both eosinophilic and basophilically stained. The next stage is late erythroblast. Its size ranges from 7 to 12 micrometer, which shows a typical cartwheel appearance of chromatin in nuclei, and hemoglobin synthesis is completed in this stage. Coming to the next stage is reticulocyte, which is the immature stage, which lacks nucleus. And the last but not the least is erythrocyte, which is the mature RBC. Its size ranges from 7.4 micrometer in diameter, which lacks nucleus and other subcellular organelles as well. So, regulation of erythropoiesis. Erythropoiesis is mediated by a hormone called as erythropoietin, which is produced from juxtaglomerular apparatus of kidney in 85% and in liver 15%. It is activated under hypoxic conditions, that is decrease in oxygen, and under anemia in decrease in RBC conditions as well. Actions of erythropoietin. It causes stem cell to differentiate, and it promotes hemoglobin synthesis by increasing the globin chain synthesis, and it induces delta alpha levulinic acid synthase, which is the rate limiting step in heme synthesis. So it releases RBC from bone marrow to circulation and causes erythroid hypoplasia of bone marrow. Factors affecting erythropoiesis. First factor is hormonal factor. Erythropoietin and androgen stimulates erythropoiesis. Thereby, men have higher RBC count compared to women because erythrocyte inhibit erythropoiesis. The dietary factors such as vitamin B12 and folic acid is required for synthesis of DNA of erythrocyte and is necessary for maturation of an erythrocyte as well. Iron required for synthesis of heme and hemoglobin. Others factors such as protein, which is required for synthesis of globin chain and hemoglobin, and intrinsic factor, which is a glycoprotein. It is synthesized from parietal cells of stomach, and it is required for absorption of vitamin B12 from gut. So, in vitamin B12 deficiency, methyl tetrahydrofolate is trapped and is called as folate trap, and homocysteine accumulates and which cause homocystinemia. Vitamin B12 deficiency and folic acid deficiency cause megaloblastic anemia. So some information about reticulocytes. It is a juvenile red cell or premature red cell. It is the first cell to appear in blood circulation. It does not have nucleus, but it has nuclear remnants which form reticular network which contain RNA, and normal reticular count is 0.2 to 2% each, and reticulocyte is stained by supravital stain which consists of brilliant chrysal blue and no methylene blue, which stains the living state of the cell, that is the RNA. And causes of reticulocytosis, that is increase in reticulocyte count, is seen in blood loss or hemolytic anemia. And cause of reticulocytopenia, that is decrease in reticulocyte count, is seen in aplastic anemia and megaloblastic anemia.